So our final chapter of the semester kind of is a piggyback off of reproductive system because we look a little bit at prenatal development. So the development in the womb before you're born, a little bit about hereditary, so the passing along of genes, and then of aging. So that's kind of what this final chapter is about. So prenatal development, this consists of the 9 months before birth, and they're a very critical part of our existence. The prenatal period is the period specifically from conception to birth, and it can be divided into 3 parts. The first 2 weeks are where we form the primitive germ layers, which will go on to form every other part of the body. And then we have from weeks 2 to 8 is when we get the major organ systems that come into existence. And then the last 7 months are really where the organ systems begin to grow and become more mature. So the embryo is the word we get to call specifically from fertilization to 8 weeks of development. The fetus is specifically from week 8 to birth. And then the clinical age is always based on the last menstrual period. And the development age begins exactly with fertilization. So 14 days after the last menstrual period. Fertilization itself is the medically correct terminology used for the union of the sperm cell and the oocyte, which is the egg, along with all the chromosomes. Capacitation makes the sperm capable of releasing enzymes to reach the secondary oocyte. The secondary oocyte will then undergo meiosis, and the zygote is kind of the resulting name for what we give to actually the fusion of the sperm and the oocyte when it contains 46 chromosomes. Remember that the sperm cell and the egg cell each only contain 23 chromosomes. So then during fertilization, when they unite, then they'll form a complete set of chromosomes, and that's what we call the zygote. So here we have, if you remember from the reproductive lecture, if you guys were able to listen to that, there's about 100 million sperm released trying to get at one egg, and whichever one kind of breaks through or comes into contact with the egg first will release its DNA into the egg. So the head of the sperm cell comes off, DNA is released, and then we have the kind of the complete fusion between egg and sperm to start kind of that zygote formation process, where 46 chromosomes have now been united to form a complete new kind of existence. So that's the zygote that results after fertilization is complete. And then this zygote begins to divide very rapidly into many, many more cells. So about 18 to 36 hours after fertilization, the zygote divides to form 2 cells. This process of cell division continues very rapidly and quickly for about 2 days. The number of cells will increase, but the size of the zygote will remain the same. And then we have kind of other names as we go through kind of the developmental processes. So we started at zygote, which was the egg and the sperm forming. Then the morula is about 2 to 3 days after fertilization. It contains about 12 to 16 cells. This will help support the placenta. And then by day 6, we change names into the blastocyst, where we'll get a small cavity to appear in the mass of cells. It will contain the inner cell mass, the stem cells, and the trophoblast. The inner cell mass portion of this blastocyst is just a thickened area. The stem cells are what will give rise to all other cell types within the body. So whenever you see the word stem cell, that will go on and differentiate to all the other cell types that the baby will need. The stem cells will be in the inner cell mass. And then the trophoblast is just what we call the remaining cells of the blastocyst. And the trophoblast is what will go on to form the placenta and the membrane that surround the embryo. So here we have the development of the blastocyst and implantation. So here we have kind of the zygote at fertilization, and then you can see by day 1, by day 2, by day 5, we are rapidly dividing cells from 2 to 4 to 12. Eventually we get to the trophoblast, and the trophoblast is what will go on and be implanted in the wall of the uterus. So the implantation of the blastocyst, excuse me, this is about 7 to 8 days after ovulation. The endometrium of the uterus is preparing itself for implantation. So the endometrium is the inner wall of the uterus, and it'll become very thick to try to get ready for potential implantation of that blastocyst. And implantation involves kind of the blastocyst attaching itself to the uterine wall and then kind of burrowing itself into the uterine wall. The trophoblast cells will digest the uterine tissue so that the blastocyst can kind of make a hole within the uterine wall. So after the blastocyst will bury itself or burrow itself into the wall of the uterus, the trophoblast cells become the chorion, which will go on to help form the placenta. And the placenta is a very important part of development. This is the structure derived from embryonic as well as maternal tissues where the fetus and the mother are attached. And chorionic villi are finger-like projections in the endometrium. Lacunae are cavities filled with maternal blood. Although you should always remember that embryonic and maternal blood supply are always kept separate, antibodies can cross over the placenta. You have nutrients and waste products must cross what we call a semi-permeable barrier between the two circulations. So some things can cross the placenta barrier, but blood cannot. So when we talk about, when we were talking about blood typing, when we talked about the Rh factor, well, the maternal blood, if it is exposed to the baby blood at birth, then it's exposed to the blood and can make antibodies. And then later on, antibodies from the mom can pass to the embryo. So that's why even moms getting the vaccine, I have a long story about trying to get the COVID vaccine while pregnant here. It's impossible, but simply put, it's okay to get vaccinated when you're pregnant, not just COVID, but for other things, because some of those antibodies can pass through to the embryo and be helpful. And we don't have to get on that vaccination talk either. I've just been going through some things trying to get vaccinated. I think I'm the only person in the world who wants to get vaccinated and I can't. Long story, I'll tell you later. The umbilical cord then is the connecting stalk between the placenta and the embryo, and it will contain all the blood vessels that carry blood from the embryo to the placenta and from the placenta to the embryo. So it's kind of the blood supply to and from the embryo. But again, we have this barrier where the two blood cells from the mom and the baby never mix. Other things can pass through, like nutrients, waste products, antibodies, but the blood can never mix or pass through. So here we have this kind of interface between a maternal and fetal circulation. We have the placenta as shown here. The placenta, again, will be connected to the umbilical cord to carry all the blood supplies to and from the fetus, the baby itself. And then here we have a look at the separation, but just between maternal and embryonic blood. So if we kind of look here, we have maternal blood filling up the lacuna spaces, kind of surrounding the placenta. But then we have this chorion layer, and that chorion layer is what disallows or not allows any blood cells to pass through between the mom and between the embryo. Maternal hormonal changes. So the human chorionic gonadotropin, this is the hormone HCG. This is secreted by that chorion layer of the placenta. It's transported to the maternal ovary, and this causes the corpus luteum to remain functional. The corpus luteum is the layer of the endometrium, so that normally if there isn't a pregnancy, the corpus luteum of the endometrium will be lost as the woman has her period. But HCG tells the corpus luteum, hey, we still need you to stick around because there's a pregnancy. So this HCG is what can be detected in a pregnancy test, in blood or in urine. It can reach its peak at 8 to 9 weeks after fertilization, but you can detect HCG in urine as early as 3 to 4 weeks, I believe, maybe right around 4 weeks. We also have estrogen and progesterone changes that are secreted by the corpus luteum that help form the placenta during the first 3 months. So estrogen and progesterone, these hormones will increase throughout the pregnancy to keep the corpus luteum and the placenta kind of alive and active. So many birth control pills are estrogen pills because what you're kind of tricking your uterus into thinking is that it's pregnant, so it doesn't ovulate you'll still have a monthly period, but that's sometimes what birth controls do. You're tricking your body into thinking it's pregnant, so you're not ovulating. So here are just some hormone concentration changes during pregnancy. High HCG levels secreted from the chorion, again, to kind of keep everything going. Progesterone and estrogen levels will also kind of be steadily increasing starting at the end of the first trimester, and then will steadily increase, throughout the remainder of the pregnancy. And again, estrogen and progesterone just keep the placenta, the uterus, kind of, active and well for the growing and developing fetus within the womb. Then we get to the formation of the germ layers, and the germ layers will go on to form other parts or other major organ systems. The amniotic cavity forms after implantation, and this will be a new cavity inside the inner cell mass of the blastocyst, and it causes part of the inner cell mass to separate as a flat disc called the embryonic disc, and that's where the embryo will grow. And the amniotic sac will be inside the amniotic cavity, and this will be filled with amniotic fluid to protect the embryo. So this is what kind of fills up the placenta. The amniotic sac is filled with amniotic fluid to help protect the embryo as well. So the ectoderm is one of the germ layers. It's the outside layer adjacent to the amniotic cavity. The endoderm is the inside layer opposite the amniotic cavity. And then the yolk sac is a third cavity within the endoderm, and we'll eventually get to pictures, I promise. The primitive streak is a structure that establishes the central axis of the embryo, and this forms when ectoderm cells migrate to the center of the embryonic disc. This is what will go on to form like the brain and the spinal cord, and this will occur or kind of be visible about 15 days after fertilization. The mesoderm then is our third germ layer, and gastrulation is what we call when cells will migrate and form this mesoderm layer, and eventually we'll go on to see what these three derm layers will go on to form. And the nodal cord is the cord-like structure that forms that central axis, and this nodal cord is what will go on to form the spinal cord. So let's look at some pictures. Embryology, we could almost have a completely separate class for a whole 16, 18-week semester on embryology. So if this is over your head and really confusing, you're probably not the only one. So as we look here we have kind of the blastocyst that's being, embedded in the wall of the uterus. We have the chorion, will be the barrier between fetal and maternal blood. The lacuna is filled with maternal blood. Here's the endometrium of the uterus is shown. And then we kind of see how this this blastocyst basically is forming. We have the amnion, the amniotic cavity, which will be filled with fluid. Here we have the embryonic disc, and remember the embryonic disc is where the embryo will eventually form. So the embryonic disc is right in the middle between kind of these three blue and yellow half circles, made up of the epiblast, the hypoblast. That's where we'll get our central streak. Our nodal cord, our spinal cord will start to develop along the central axis. The yolk sac, the blastocele, will surround this inner cell mass. And from there, we start to form the embryo, basically right within this embryonic disc layer. So here we have the primitive streak and the germ layer starting to form. So here we have, again, the embryonic disc refers to the central part, and that's where the embryo will begin to form. And these are the three germ layers. We have the ectoderm, the mesoderm, and the endoderm that are forming. And in order, and here we have the caudal end, which will be the tail or the rump. And then the cephalic end will be the head of the embryo. The cells, so number one, let's read through kind of how these form one at a time. The cells in the surface epiblast move toward the primitive streak, so you can see this showing here, and migrate through the streak, which are shown by our blue arrow tails. And then cells of the epiblast that migrate through the primitive streak become endodermal and mesodermal cells. So those are then the red arrows. And the mesoderm in pink will lie between the ectoderm in blue and the endoderm in yellow. And we'll go over what each germ layer will go on to form. But in general, the ectoderm is forming our nervous system, our nervous layer. So that'll go on to form the brain and spinal cord. with the mesoderm and endoderm kind of forming everything else. So endoderm. So endoderm, we're going to work our way at the bottom first. So what does the endoderm go on to form? Lining of the digestive tract, the lungs. So a lot of linings, lining of the hepatic, pancreatic, and other ducts, the thymus gland, the thyroid gland, parathyroid glands, and tonsils. The mesoderm, if you can remember, mesoderm muscles. So the mesoderm forms the dermis of the skin, heart and blood vessels, kidneys, gonads, muscle, bones, all except your facial bones, and microglia, which are kind of supporting cells, glia cells of the nervous system. And then the ectoderm also forms the outer layer of the skin, so the epidermis, tooth enamel, the lens and cornea of the eye, nasal cavity. The ectoderm is what will form your brain and spinal cord, which is a big one. Melanocytes are the cells that go on to produce melanin to protect against harmful UV rays, and the adrenal medulla, which is a part of the adrenal gland. So the neural plate and tube. So this is what forms about 18 days after fertilization. It's when the ectoderm overlying the nodal cord thickens. So we're talking basically about like a fold where the edge of the neural plate will rise and come together. And this kind of fold, when they come together, will eventually form a neural tube. It's called the neuroectoderm, and this will what will go on to become the brain, the spinal cords, and part of your peripheral nervous system. So we see right here the neural plate. We have a groove forming. We have folds kind of coming up on either side of the groove. Eventually, they'll come together to kind of form like a tube in the middle. And then the nodal cord will form below it, and that'll go on to form the brain and spinal cord. So that's the formation of the neural tube. General body structures, so limb buds. You can see limb buds around day 28. I think you guys know I'm pregnant. I can't remember which classes I've told, but I think I was like 9 weeks pregnant, and you could already see in the ultrasound like little arms and legs, really just like limb buds. So by day 28, you could see those. By day 35, the zones of cells are dying between your fingers and toes so that you can have separation. And the face will develop by fusing 5 growing masses together called processes. So this is a look at a human embryo about 35 days after fertilization. You can see the heart beating, the eye, you'll see this nodal cord or, the spinal cord developing, and then the limb buds shown as well. Development of the face is kind of interesting. It's about 28 days after fertilization. You can see these processes kind of coming fused together with a nose beginning to form about 33 days. 40 days, more processes are coming together to form upper limbs and lower limbs. And then by about 14 weeks, you can really kind of start to see the eyes. They're not open, they're closed, and the lower lip and jaw. So that's kind of how the development of the face comes in. Organogenesis is when the organ systems develop between weeks 2 to 8. The GI tract begins forming at about day 28, and the heart develops from 2 blood vessels about day 21 and already starts to beat at about day 21. Here's the digestive tract forming. So again, you can kind of see the digestive tract forming from the endoderm, again, forming like another tube-like structure. So this is kind of how we're getting from the primitive streak, which again is right in the middle of these 3 germ layers in blue, kind of pink, and in yellow. And in yellow, the endoderm is where your digestive tract will form. Embryonic, digestive, and urinary system. You can see how the kidneys are forming. You can see some of where the other limbs are forming, again, coming off of that primitive streak area. Formation of the heart is about 20 days after fertilization. The heart consists of these 2 parallel tubes, and eventually the tubes will form kind of one tube and ventricles will form. Then right and left atria will form. And the final embryonic condition of the heart is shown here. You can see the 4 chambers, but you see a hole in between the atria. That hole is really important so that blood bypasses the ventricles and goes right into the left atrium. The embryonic heart doesn't really need blood in the ventricles at all because it doesn't need to pump blood to the lungs to receive oxygen because the lungs are not fully functional yet. So that's why we have this hole in the interatrial septum. That usually closes or slams shut with a baby's first breath due to differences in pressure. It's the fossa ovalis is the remnant of that hole. Growth of the fetus, the embryo becomes a fetus about 8 weeks. The beginning of the fetal period is marked by the beginning of bone ossification. So as soon as the bones start to harden and become ossified, at about 8 weeks, then we kind of change names into the fetus. And then in the embryo, most of the organ systems are developing, whereas in the fetus, the organs are just present. And during the fetal period, the organ systems will enlarge and mature. So here's a late embryo and then the fetus, obviously beginning to mature more and more. Parturition then is the process where a baby is born, usually occurring at 14 weeks. The uterus, made up of smooth muscle, starts to contract involuntarily without you telling it to. The cervix thins and dilates, so it becomes larger to eventually open into the birth canal. And then these are the 3 stages of labor. The dilation phase is when the cervix dilates up to 10 centimeters. It takes about 24 hours. I mean, that 24 hours, it can be 2 hours, it can be 36 hours. The amniotic sac will rupture, and that's what causes the water breaking or the amniotic fluid within the sac to break. So that's what would exit out of the female. The second stage is the expulsion phase, where the cervical dilution is complete and the baby exits. And then the third stage, some people forget about this, you actually have to push out the placenta as well. So after you push out the baby, and by then you're just happy that the baby's there, you still have to push out the placenta as well. And then you can turn your placenta into pills and take placenta pills. There's some nutrition benefits to that. I don't know if anyone has done that in here. I have not, but some women do that as well. They eat their placenta in the form of pills. They can change it into that. Again, not trying to start an argument, just wanted to let you know. Okay, so this is parturition, the 3 stages. First stage, the cervix dilating, the water breaking, amnion ruptures, amniotic fluid leaks out. The second stage is when the fetus is expelled. And then the third stage is actually pushing out the placenta as well. Factors influencing parturition. So what makes a woman go into labor? The fetal hypothalamus will actually release a hormone that stimulates ACTH secretion from the pituitary. The fetal pituitary secretes ACTH in greater amounts during or closer to delivery. ACTH will cause the fetal adrenal cortical glands to secrete greater quantities of adrenal cortical steroids, which will travel through the umbilical cord through the umbilical blood to the placenta. And in the placenta, these fetal cortical steroids cause progesterone synthesis to level off and estrogen and prostaglandin synthesis to increase. And this will make the uterus more excitable. The uterus will begin stretching, producing action potentials that are then transmitted to the brain of the mother. And these action potentials then will stimulate the mother to secrete oxytocin. And oxytocin is the big hormone secreted from the posterior pituitary of the mother that causes the uterus to contract. And oxytocin is one of those positive feedback hormones. So oxytocin will continue to increase to whatever amount it needs to until the baby is born. All right, so the newborn respiratory and circulatory changes. So forced gasps of air occur right when the baby is born. And when it does this, the lungs will inflate. And this is why it's very important for babies to be born after 38 weeks, because at 38 weeks, the lungs have those cells that are producing surfactant. And surfactant is what will keep the lungs inflated. So once the baby takes its first breath, the change in pressure causes the lungs to inflate, and then they'll be kept open by the surfactant. Before 38 weeks, it's a really close time frame. Before 38 weeks or 36 weeks, I should say, those lung cells aren't producing adequate amounts of surfactant. So even a baby born 36 weeks might have some respiratory problems and just need to be carefully watched until their lungs can produce that surfactant, which again is like a fluid that lines your alveoli to basically keep them inflated like a balloon. The changes in the lung pressure at that first breath lead to closure of the foramen ovale and the separation of the heart into 2 pumps. So once that foramen ovale closes, which is the hole between the atria, in the wall between the atria, Then blood will begin to go down into the ventricles so that blood can now be pumped to the lungs because the lungs are inflated and ready to accept oxygen. The ductus arteriosus closes, which is another hole that travels from the pulmonary trunk to the aorta. So it's another place to just bypass blood going to the lungs before a baby's lungs are ready. And then the umbilical cord needs to be clamped because that causes closure of umbilical veins and arteries, leading to the closure of the ductus venosus, another hole in the heart. So that's why you need to clamp or cut the umbilical cord right away. So here is circulation in the fetus. So blood will bypass the lungs by flowing from the pulmonary trunk through the ductus arteriosus to the aorta. So this is the ductus arteriosus hole for blood to bypass going to the lungs to go right into the aorta. Number two, blood also bypasses the lungs by flowing from the right to the left atrium through the foramen ovale hole. And again, that hole will close when the newborn takes its first breath. Blood passes by the liver, bypasses the liver by flowing through the ductus venosus. So blood won't even go to the liver yet. Oxygenated blood from the placenta will be passed to the fetus by the umbilical vein, and deoxygenated blood is carried from the fetus to the placenta through the umbilical arteries. So the fetus is still getting oxygen and getting rid of waste through the umbilical cord, through the umbilical artery and umbilical vein. And then what happens when air enters the lung? Blood is forced through the pulmonary arteries to the lungs. So the ductus arteriosus closes, and usually you can still see a remnant of the ductus arteriosus as a little piece of tissue in a cadaver. Normally it would be a hole there, but now in us, it'll just be a remnant. The foramen ovale closes, so blood can no longer flow from the right to the left atrium. The ductus venosus degenerates and becomes a ligament, basically, shown here, 3, which will now be able to, so blood will go to your liver now. And then the umbilical arteries and veins are cut when they cut the cord, and the umbilical vein becomes the round ligament of the liver, and the umbilical arteries also just degenerate. So it's interesting how, everything becomes a part of the liver or then goes on to have a purpose. So that's a little bit of circulation in the newborn. Obviously, there's problems a lot of times with, these holes not closing properly. Blood is still bypassing or going from atria to atria. If this hole didn't close all the way, that can be a problem because you want that hole to close completely so the heart can function at maximal capacity. So the ventricles are doing the pumping and blood isn't flowing any other way. Some digestive changes in the newborn. Meconium can be present in the amniotic fluid. It's a greenish waste containing mucus from the GI tract and bile from the liver. So this is basically just waste that the fetus is producing. So, childbirth is kind of a messy process because everything has to be removed. The neonate usually loses about 5 to 10% of its total body weight during the first few days of life. So usually newborns will kind of dip down in their weight, and then eventually it will become capable of digesting lactose. Lactation is the production of milk by mammary glands. Estrogen and progesterone levels will fall right after delivery, and prolactin is the hormone that's then released to stimulate milk production. Colostrum is what is released first by the mother as a high protein pre-milk, which contains a lot of antibiotics. So if the mother is deciding to nurse, getting this colostrum, which might not seem like a lot, it's just a little bit, that comes out via the mammary glands, but it has a lot of proteins and antibodies. So hormonal control of lactation. So if any of you guys have breastfed, you probably, will know what I'm referring to here. And if not, it's okay, but stimulation of the nipple by the baby's suckling will actually initiate an action potential that goes through sensory neurons that go to the hypothalamus. And in response, the hypothalamus releases stimulates the posterior pituitary gland to secrete oxytocin, and the anterior pituitary to release prolactin. What oxytocin does is oxytocin lets down the milk, and prolactin continues milk production. So actually the sucking of the baby, the woman usually isn't just leaking milk all the time, sometimes at the beginning, but usually the baby sucking will then allow the milk to come. And then prolactin keeps the milk being produced in the mammary glands. So life stages, again, as we review them prenatally, and then we'll go on to postnatally, the germinal period is fertilization to 14 days. The embryo is what we call 14 to 56 days after fertilization. The fetus is 56 days after fertilization to birth. And then the neonate is the name we give for about 1 month to 1 to 2 years after birth. Infant is kind of another word for neonate, 1 month to 1 to 2 years. Child is what we specifically would call, 1 to 2 years to puberty. Adolescent is puberty to 20 years, and then an adult is 20 years to death. So those are how we kind of divide out the life stages. So aging. So cell division occurs rapidly during early development, and then it slows with age. Neuronal loss will be greatest early in life, and neurons aren't replaced. Collagen becomes rigid and less flexible. Skeletal muscle fibers will peak around 20 to 35 and then decline with less activity. When do we see most Olympic athletes? 20 to 35? 35 is probably even pushing it. But if you think of other major sports athletes, their skeletal muscle fibers are peaking about 20 to 35. And then you'll get athletes who can go on forever, which is probably their own choice. It's probably hard to retire from something like that. Your cardiac muscle cells will lose elasticity, blood flow decreases, and then death will occur to all of us. This is a permanent cessation of life functions, and causes can vary. We know, your heart can stop beating through a variety of different mechanisms. Sometimes your heart can stop beating and you're the healthiest person out there. You don't have any atherosclerosis or clots, but your heart can stop. Your kidneys can stop. Your kidneys failing also will quickly lead to death because your kidneys filter all of your blood. And then brain death is the medically, I believe the medically correct term for kind of saying someone is dead when there's no electrical activity going on in their brain. So we just have a couple slides to get past through genetics and a little bit of hereditary. So genetics itself is the study of hereditary. So the ability to pass on your genes to the next generation. Chromosomes are genes with DNA. Chromosomes, all cells except your sex cells have 46 pairs. So all of the cells that replicate and divide in the body have 46 pairs of chromosomes. Your sex cells, the egg and the sperm, they each have 23 because their goal, or maybe hope one day, is to form a new 46 complete pair. The karyotype is a visual display of all chromosomes that's useful for looking at some chromosomal abnormality. And then here are the autosomal chromosome pairs. We have 22 pairs. They're all chromosomes, but the sex chromosomes. And I'll show you what a karyotype looks like in the next picture. Females have 2 X chromosomes and males have 1 X and 1 Y. Gametes is another word for a sex cell, the sperm and the egg. These are always produced by meiosis. So most all cells in the body are duplicated and reproduced by mitosis. Like gametes kind of go through 2 divisions because they result in just a 23 set of chromosomes in each. Homologous chromosomes are chromosomes that contain the same complement, genetic information from each parent. An X sperm will produce females and a Y sperm will produce males. So if you're wondering who gets the say, whether you have a boy or a girl, it's always the sperm that decides. The sperm will bring an extra X to the table and it'll be a girl, or the sperm will bring an extra Y to the table and it will be a male. So here's the human karyotype. It's a visual display of the chromosomes in a somatic cell. There will always be 23 of autosomes, which are all the chromosomes except the sex chromosomes. And then there will be 1 pair of sex chromosomes. And I'll show you what I mean by that. The sex chromosome pair of a normal female is XX, whereas the sex chromosome pair of a normal male is 1X and 1Y. And again, we can run into some issues, which we'll talk about too. So here we have the human karyotype, which displays the chromosomes. So each chromosome is kind of a pair. And you can see there's 22 kind of normal autosomal chromosomes. And then the 23rd pair will be the sex chromosome. And this is a female because it's an XY, or a male, excuse me, because it's an XY. and a female would be XX in this 23rd sex chromosome pair. The inheritance of sex is always a 50-50 chance. So every time, someone gets pregnant, there's always a 50-50 chance that it could be female or male. And that is what is shown here through meiosis, the oocytes, what they will bring, and then through meiosis, the sperm cells, because the female is XX and the male is XY. It's really the male who then will determine that extra X will make it female, or the Y will make it male. So that always comes from the male side of things, but it's always a 50-50 chance. So people can have 10 girls, they can have 10 boys, and just that's how it is. That would be a lot of kids. Genes are the functional unit of hereditary. They consist of a portion of DNA. So a gene is just a portion of your entire DNA. Alleles are similar genes on homologous chromosomes. And if you are homozygous for a particular trait, you have two alleles that are identical. And if you're heterozygous for a trait, you have two alleles that are different. And I'll show you a little bit what I mean by that. Dominant and recessive genes. A dominant gene is a gene that is expressed, and this is written always as a capital letter. And a recessive gene is a gene that is not expressed, and this is written as a lowercase letter. And they've gone through and figured out what are dominant genes and what are recessive genes. So for example, a recessive trait is having red hair. So my daughter has red hair, but my husband and I do not. So the only way that she can express that trait of red hair is she received two recessive genes, one from me and one from my husband. And she has the two recessive genes put together, and then she can express that recessive trait. Otherwise, the dominant trait will always be expressed. The genotype are the alleles a person has for a given trait. So the genotype will be kind of the genetic code for a given trait. And the phenotype is what we describe a person's appearance. And again, we're not getting into this in too much detail. So if you're lost already, don't worry too much. The pedigree of a simple dominant trait, what this is showing, so you can look at your pedigree or your family history for different traits. And this is showing, I can't remember if the square or the circle is male or female, but it's showing how dominant and recessive traits get passed on. And I believe, I think the dominant trait is orange and the purple trait is recessive. I can't remember, but what it just goes to show is how different traits, whether they're dominant or recessive, will get passed through the generations. And you can go back and do your own pedigree of a simple dominant trait to see if it was expressed or not. You can do red hair. Another trait you can do is whenever you kind of fold your hands, if your left thumb is over the right, that's recessive, or if your right thumb is over the left, that's dominant. Other types of dominant, every trait they've figured out if it's a dominant or a recessive trait. A sex-linked trait is always a trait affected by genes on one of the sex chromosomes, specifically on the X-linked. So for example, if someone has hemophilia A, which means a person lacks certain clotting factors, this would be a trait that's linked on their sex chromosome, the X-linked. So this is a look at hemophilia and how it can get passed on. If you guys haven't, really worked with Punnett squares yet and how things get passed on, what this is showing are the different combinations of passing on a trait, for example. So here's a mother who's a carrier for hemophilia, and the carrying the hemophilia is represented by this small h. So the mother can pass on the trait for hemophilia to her offspring. Looks like there's a 25% chance. And then the husband is normal, or the father is normal, and he will just pass on his normal XY chromosomes, but he is normal. So the big H means he does not have hemophilia, nor does he carry it. So the mother, she doesn't have hemophilia either, but she's carrying the trait for hemophilia. So she could pass on Again, hemophilia is a sex-linked trait because it's a trait that's passed along her X chromosome. And that hemophilia trait, even though she's normal, she can still carry it and pass it along to offspring. And then the genotype of a chance that, the only looks like all the possible combinations, the only way that someone could have hemophilia is if they have the little h. But remember, if there's the big H, that means they're normal. And the big H will always kind of overseed the little h. So this female down here, she will be normal, but she'll carry the H, the hemophilia trait, to pass on to her offspring. The male, however, so there's a 1 in 4 chance that if they have a male, the male could be born with hemophilia. So this is just showing how hemophilia, it's a blood clotting disease. It can be passed on with someone who doesn't have it because it's still linked to their trait that could be passed on to an offspring. Okay, a little bit more about genetics, and I promise there's just a couple slides left on this. It's a great way to end the class talking about genetics. If this is really interesting to you, go on and keep taking more classes like this, because genetics is interesting and there's a lot of good things coming out of genetics now. So incomplete dominance is a genetic condition in which a dominant allele does not completely mask the effects of a recessive allele. So they might kind of both be expressed. Codominance is a genetic condition in which 2 alleles combine to produce an effect without either of them being dominant or recessive. A polygenic trait are those traits determined by the expression of multiple genes on different chromosomes. And then we can get to some genetic disorders. They can be caused in any abnormality in someone's DNA. And the interesting thing about a genetic disorder, it could involve like a change or a mutation in a single gene or even a single base pair on the DNA strand. So a mutation specifically is a change in a gene that usually involves a chain in the nucleotide composing the DNA. So it's a really teeny, minute change in one person's DNA in one of the base pairs that can go on and cause a genetic disorder. Genetic counseling includes predicting possible results of matings involving carriers of harmful genes and talking to parents or prospective parents about the possible outcomes and treatments of a genetic disorder. This can be helpful because some of these X-linked traits, for example, I think it's Huntington's disease, it's not expressed in an individual until they're 40 or 50, and most likely they could pass that on to their offspring. And most likely they might not, but just with this knowledge, prospective parents can make informed decisions about having children. The first step in genetic counseling is to attempt to determine the genotype of the individuals involved. They usually take a family tree or a pedigree to look at historical information about family members. Sometimes knowing phenotypes of relatives even makes it possible to determine a person's genotype. A part of the process is collecting information. A karyotype can be prepared, which is, again, the visual representation of chromosomes. And for some genetic disorders, the amount of a given substance, such as an enzyme, can even be produced by a carrier that can be tested. So that is where we'll end with chapter 20 and genetics. So I'll go ahead and end the recording, and then I will answer any questions